Right now, you're a miracle of order, atoms resisting a universe that's built to fall apart. Your heart beats, your cells negotiate chaos, and yet physics can't point to a single testable reason you, specifically, are alive in this exact moment. So what is life, cosmologically, and what does it mean if the universe has no explanation for why you're here at all? In the laboratory, alive is not a spell we can cast. It's a pattern we can interrupt. And when the pattern breaks, nothing in the equations tells us where you went. Biology can list the parts, name the pathways, and narrate the chemistry with clinical precision. Yet the category itself keeps slipping. Between a crystal-like virus and a dormant spore, between a sterile worker and a self-copying flame, the border turns ambiguous, then political, then strangely personal. Because what you call a body is less a structure than a negotiated truce. Membranes hold gradients, enzymes pay constant repair bills, and errors are tolerated only within a narrow budget. The system persists by measuring the world, correcting itself, and exporting its disorder, an information engine wearing matter like clothing. But information has more than one alphabet. Some of it arrives as RNA, some as integrated viral history, and some as shape, conformations that replicate without genes, rewriting the meaning of inheritance. The unsettling implication is not that you are fragile, but that self is porous by design. Then the camera pulls back. If our instruments are tuned to one familiar chemistry, what else could be thriving unnoticed, even here? Because we never learned how to ask the right questions. And if life is a threshold event in the universe's flow of energy, what decides when that threshold becomes a witness, rather than a mere reaction? Before we begin, let us know in the comments below where in the world you're watching from and what time it is there. Now settle in for a journey. Across 4 chapters, we follow life from broken definitions to foreign code, then outward to possible hidden biospheres and cosmic constraints. By the end, the mystery sharpens into a disturbing possibility. Science may explain every mechanism and still not possess a universal reason for you. You are a temporary island of order in a universe that prefers disorder, a brief pattern standing against an immense statistical tide. Biology can catalogue your parts with exquisite precision: cells, membranes, enzymes, genes, yet it still struggles to state what makes them alive. After 150 years of modern biology, there is no universal operational definition that cleanly separates living from non-living across every case. The problem is not philosophy, at least not primarily. It is measurement, prediction, and the practical need to recognize life when we might see it only once. Cosmology sets the pressure, because the arrow of time is written in thermodynamics. The universe trends toward higher entropy, and order decays unless work is done to maintain it. In plain terms, organized structures tend to spread out, break down, and drift toward equilibrium. Life persists by maintaining local order, while exporting entropy to its surroundings: heat, waste, and chemical byproducts leaking outward. In a thermal camera, a body is a bright plume of dissipation, constantly paying energy to keep its internal patterns intact. That survival trick is clear, yet it raises a sharper question. What exactly is being maintained that we call aliveness, and why does it hold together now? Even at the cellular scale, the bookkeeping is relentless. Cells pump ions against gradients, repair damaged proteins, and replace molecular parts that would otherwise unravel. The apparent stability is an illusion produced by continuous turnover, like a whirlpool that keeps its shape while the water changes. If life is a kind of organized flow, then the boundary between thing and process becomes strangely unstable. And if it is a process, what features must that process include to count? In the 1940s, Erwin Schrödinger tried to connect life to physics with a simple phrase that still echoes. Life feeds on negative entropy, or negentropy, by drawing usable order from the environment and delaying equilibrium. He was writing in the shadow of early quantum theory, when physicists were newly confident that deep principles could unify disparate phenomena. The framing explains how organisms avoid equilibrium for a time, and why they must constantly consume energy. It does not explain why some organized systems count as alive while others do not, even when both resist disorder. So biology tries boundaries, attempting to build a checklist from observable traits. Metabolism seems essential. until dormant spores and frozen organisms blur the line with near-zero activity. Some microbes can sit in stasis for years, then revive when water and nutrients return, as if time had been paused. Reproduction seems essential, until sterile workers in insect colonies still live and function within a larger reproductive machine. Homeostasis seems essential, until some parasites outsource regulation to hosts, borrowing stability instead of generating it. Evolution seems essential, until individuals live without evolving, and evolving systems can be built from non-living chemistry under the right conditions. Each definition fails in two directions, and the failures are instructive rather than embarrassing. It excludes something we intuitively call alive, or it includes something we do not want in the category. The edge cases are not rare exceptions tucked into footnotes. They are stress tests that reveal the definition is doing the wrong kind of work, like a net woven for one ocean and dragged through another. In 1935, Wendell Stanley crystallized tobacco mosaic virus, and the images still feel like a provocation. In glassy crystals, the virus sat in a rigid lattice, stable and inert, behaving like a chemical that could be weighed and stored. Under a microscope, it looked less like an animal or a plant and more like a repeating geometric artifact. Yet inside a host it switched roles, hijacking cells and producing more of itself with ruthless efficiency. It exploited ribosomes, enzymes and raw materials, turning the cell into a factory for viral copies. The result forced a question biology still cannot retire. Is life a substance you can bottle, or a process that only exists in motion? That question matters because modern agencies need working rules, not perfect ones, especially when designing instruments and missions. NASA offers a definition that is meant to be operational rather than poetic. Life is a self-sustaining chemical system capable of Darwinian evolution. It is practical for missions, and it is fragile under pressure from real biology. Sterile organisms like mules are alive, yet they fail the evolution clause as individuals, even though their lineages arise through evolution. Fire can look self-sustaining and self-propagating, and it can spread across landscapes like a hungry organism. Some autocatalytic chemistries can mimic growth, consuming feedstock and expanding patterns without heredity that stores detailed instructions. The deeper issue is that our test often targets surface behaviors instead of underlying organization. A rover can measure gases, isotopes, or complex organic molecules, but those signatures can be ambiguous. Chemistry can imitate biology, and biology can hide behind dormancy, scarcity, or unfamiliar solvents. When the definition wobbles, the instruments inherit that uncertainty, and every detection becomes a debate about thresholds. Now the tension widens, because the stakes are cosmic rather than academic. If we cannot define life cleanly on Earth, how do we search for it in the cosmos with confidence? The Great Filter and other cosmic theories depend on what counts as life in the first place, and where the critical transitions begin. Is the filter about the origin of metabolism, the rise of heredity, the leap to complex cells, or the emergence of intelligence? An unstable definition turns every estimate into a moving target, and every silence from the sky into an unclear signal. So we change the angle, not to escape the problem but to make it measurable. If life is not defined by what it is, maybe it is defined by what it does, how it stores patterns, copies them, and corrects errors while the universe pulls it apart. We follow the trail from matter to code, from chemistry to information, replication, and control. And we ask, with new seriousness, whether alive names a substance at all, or a particular kind of organized persistence. So we change the angle. Instead of asking what life is, we ask what life runs. Picture the camera drifting inward, past skin, past capillaries, past the soft turbulence of blood, until the world becomes membranes and ions. Red cells slide like coins through narrow passages, and white cells roll along vessel walls sampling chemical cues. At that scale, a living cell is not a tiny person. It is a cybernetic loop embedded in chemistry and thermodynamics, and it survives by continuously steering matter and energy. The boundary is a lipid bilayer only a few nanometers thick, two molecular sheets whose oily cores repel water and trap charge. That thin film is not decoration. It is the physical reason a cell can maintain differences between inside and outside, and differences are where usable energy hides. A cell senses gradients in salt, sugar, oxygen, and temperature, then converts those differences into internal signals. receptors change shape when a ligand binds, and that shape change becomes chemistry, a cascade of phosphorylation, second messengers, and altered gene expression. Those signals are not thoughts, but they are state, molecular marks that bias what happens next. The cell acts on the world by opening channels, pumping ions, building polymers, and changing shape, and it repairs itself because everything in it is always decaying. Proteins unfold, lipids oxidize, and DNA suffers tiny insults from water and reactive oxygen. Even without outside threats, the cell is a high-speed maintenance project that never finishes. Energy flows through the cell like a river through a mill, and information steers that flow toward persistence. In biochemical terms, the cell couples exergonic reactions to endergonic work, often using ATP as a transferable currency. ATP is not magic fuel. It is a molecule whose phosphate bonds can be traded again and again to push unfavourable reactions forward. In plain language, it spends fuel to keep its structure from falling apart. It spends fuel to keep sodium out and potassium in, because charge separation is a battery. It spends fuel to keep proteins folded and membranes patched, because equilibrium would be dissolution. The loop is not mystical. It is control under noise, maintained far from equilibrium, and it is always one interruption away from collapse. If gradients vanish, if membranes leak, or if catalysts break faster than they are replaced, the loop unwinds into chemistry that no longer corrects itself. This reframing sounds clean until we inspect the machinery that makes it work, because the machinery is both elegant and indifferent. Biology often speaks as if DNA contains a plan, and the cell follows it, as though a genome were a blueprint read by a careful builder. That metaphor helped early genetics feel intuitive, and it still shapes how we talk about instructions. The closer view is stranger, because the cell is a chemical computer that executes instructions without knowing what they mean. It does not hold a model of the whole organism. It holds local rules, local interactions, and a history of what survived. Even the word instruction is a metaphor that can mislead us. Molecules do not understand goals. They collide, bind, change shape, and fall apart, and out of that blind traffic a stable loop can emerge. At the centre sits the ribosome, a universal translation engine found in every known cellular lineage. Its core is ancient RNA, a reminder of a deep evolutionary past when RNA likely played both information and catalytic roles. Visually, it is a dense, asymmetric machine. Two subunits clasped together around a thread of RNA. It reads RNA sequences in triplets, called codons, and links amino acids into proteins with rhythmic precision. The ribosome advances in discrete steps, like a ratchet, and each step depends on exact geometry at the decoding site. In the background, GTP is spent as a timing and accuracy mechanism, because even molecular machines need a clock. The mapping between codons and amino acids is the genetic code, shared across almost all known life with only minor dialects. That near universality is one of biology's strongest clues that all modern cells share a common ancestry. It also means a message written in one lineage can often be read in another, which is a gift to biotechnology and a vulnerability to infection. In the foreground, transfer RNAs act like adapters, each carrying a specific amino acid and matching a specific codon by base pairing. The anticodon loops probe the mRNA, while their other ends present the amino acid like a standardized plug. The ribosome does not interpret. It performs a physical procedure that turns one polymer pattern into another. Translation is execution without comprehension, and the indifference is literal. The ribosome does not ask whether a protein will help the cell, and it cannot anticipate whether the product will be toxic. It does not check whether an RNA message is native or foreign, because native is not a chemical category. It does not verify that the resulting protein will fold safely into a stable structure. Folding is governed by physics, hydrophobic collapse, hydrogen bonds, and electrostatic interactions. So the ribosome merely releases the chain, and the chain takes its chances. Chaperone proteins may intervene, offering sheltered spaces for folding, but even chaperones do not know function. It simply runs the mapping driven by binding energies, kinetic rates, and a supply of charged tRNAs. If the supply changes, translation slows. If the temperature rises, mispairings become more likely. The machine keeps going, because stopping is not an option built into chemistry. If we could slow time, we would see a repeating choreography. An mRNA strand feeds through a narrow channel, codon by codon, while tRNAs arrive like couriers docking and departing. Each successful match triggers a conformational shift, and each shift ratchets the machine forward. The growing protein chain snakes through an exit tunnel, hidden inside the ribosome's body. Outside, the cytoplasm is crowded, less like a dilute soup and more like a bustling city of macromolecules. In that crowd, diffusion is slow, collisions are constant, and order must be actively maintained. That indifference is not a flaw. It is the condition that makes biology scalable, because the same translation engine can build an astronomical variety of proteins. A machine that can translate any message can build any protein, if the message arrives and the amino acids are available. The cost is that meaning is external to the machine, and that is the unsettling part. Meaning is imposed by selection, error correction, and survival, not by a semantic layer inside the ribosome. The ribosome is a service, and services can be used. So the question shifts again. If life is code, where does truth come from? The answer begins with error rates, because copying is never perfect in a thermal world. At the molecular scale, heat is not background ambience. It is relentless jostling that bends bonds, shakes base pairs, and occasionally flips a nucleotide into the wrong partner. Cells rely on proofreading, redundancy, and repair to keep information coherent, even though every bond is vulnerable to heat and chemistry. Polymerases copy DNA with astonishing fidelity, yet that fidelity is engineered by mechanisms that cost energy. Accuracy is purchased, not granted. DNA polymerase selects nucleotides by shape complementarity and hydrogen bonding, and many polymerases also proofread with a second active site. When a mismatch slips in, the enzyme can backtrack, excise the wrong base, and try again. The pause is visible in single-molecule experiments, a tiny hesitation that prevents a permanent error. Even then, damage accumulates from oxidation, radiation, and spontaneous chemical changes. Cytosine can deaminate into uracil. Bases can be alkylated by stray chemicals. A double helix is stable, but it is not invulnerable, and time is always working against it. Repair systems patrol the genome like maintenance crews moving through a city at night. They recognize distortions in the helix, excise mismatches, and patch breaks using the intact strand as a reference. Some pathways fix single base errors, while others rejoin double-strand breaks that could otherwise shatter chromosomes. The details matter because repair is not one thing. There is base excision repair for small lesions, nucleotide excision repair for bulky distortions, and mismatch repair for replication slips. Each pathway is a different toolkit, tuned to a different kind of molecular accident. Translation has its own quality controls, but they are statistical, not semantic. Aminoacyl tRNA synthetases must attach the correct amino acid to each tRNA, and they use editing pockets to reject near misses. This is one of the quiet miracles of life, because a single mischarged tRNA can mislabel many proteins. The ribosome itself checks geometry at the decoding site, yet it cannot know whether the message is beneficial. It can reject mismatched shapes, but it cannot reject bad ideas. The distinction is sharp, and it runs through every cell you have. The system works because errors are usually rare enough, and because selection removes many catastrophic outcomes after the fact. In a population, lineages that drift too far from viability vanish, leaving behind those whose error budgets stay within limits. Truth, in this sense, is not a guarantee at execution. It is a filter applied by reality. This is why information in biology is not like information in a mind. It is not a statement with guaranteed reference, and it is not validated before it acts. It is a pattern that tends to produce a viable next state, given a particular environment and a particular history. When the pattern fails, the cell does not notice in advance. It fails in reality by misfolding a protein, rupturing a membrane, or losing control over replication. The feedback arrives as death, sterility, or competitive loss, and that feedback is brutally honest. The central dogma is often taught as a foundation, and it deserves that status as a map. DNA makes RNA. RNA makes protein. It organizes a century of experiments, from bacterial genetics to molecular medicine, and it tells us where to look when we want to change a trait. Historically, that clarity was hard-won. Early 20th century biologists argued over whether proteins or nucleic acids carried heredity, and the answer emerged through painstaking experiments and bitter debate. The dogma became a compass because it simplified a bewildering landscape into a direction of flow, but it is not an explanation for why the flow coheres into an organism instead of chemical noise. A pipeline can exist without producing a stable product, and a stream of transcripts can exist without becoming a body. The dogma describes directionality, not governance. The dogma does not explain why some sequences become stable heredity rather than transient molecules. It does not explain why a cell maintains boundaries and identity through constant turnover, even as its components are replaced. It does not explain why a genome becomes a body rather than a pile of transcripts. It describes a pipeline, not the reason the pipeline holds together. The missing piece is the control architecture that keeps the system within viable ranges, despite fluctuations and intrusions. In other words, the dogma tells us what happens, but not why it remains coherent. To see the crack, we look at what the pipeline will translate if given the chance, because translation is permissive. The ribosome is not a gatekeeper, it is a service. Viruses exploit that service with brutal elegance, and the visuals almost write themselves. Outside a host, many viruses are inert packages, protein shells around nucleic acid, sometimes wrapped in a stolen membrane. Under the microscope, they look like geometry, like tiny capsules and spikes, built for attachment. They do not metabolize, they do not repair, they wait. In the dry light of a lab bench, they can be crystals of potential, stable enough to persist until they find a living cell to run them. Inside a host, they become active information, redirecting cellular resources toward viral replication. They bring sequences that the cell will translate, even when those sequences are not the cell's, and they often arrive with signals that mimic cellular messages. A ribosome cannot tell whether an mRNA came from the nucleus or from a viral genome. The viral strategy is not to build a new machine from scratch, it is to hijack an existing one, because the host already maintains the gradients, the nucleotides, and the ribosomes. In a sense, the virus is a minimal program that assumes the hardware is already running. Some viruses shut down host translation, or selectively favor viral messages by manipulating initiation factors and RNA structures. Others hide their RNA from detection by capping it, modifying it, or replicating inside membrane compartments. These tricks are not intelligence. They are evolved compatibility with a system that never asks why. Retroviruses go further, and historically this was one of the most conceptually disruptive discoveries in molecular biology. They copy RNA into DNA using reverse transcriptase, an enzyme that runs the central dogma backward. That reversal was once unthinkable, because it suggested the map was not the territory. That DNA can integrate into the host genome, becoming part of the chromosome itself. Integration is not gentle. Viral integrase cuts host DNA and stitches in the new sequence, leaving behind scars that can be traced long after the infection is gone. If integration happens in germline cells, the viral sequence can become hereditary. An infection becomes an inheritance, and the boundary between self and invader shifts across generations. What was once an acute event becomes a permanent annotation in the genetic record. In humans, this is not a rare curiosity. Roughly 8% of the human genome is derived from endogenous retroviruses, ancient infections fossilized in DNA. These sequences are foreign authorship embedded in what we call our genetic code, and they sit among genes like old graffiti that sometimes becomes architecture. The camera can linger on a genome browser view, rows of letters punctuated by repeated motifs, while a timeline stretches back through primate evolution. Insertions accumulate like sediment layers, and each layer marks a moment when an external replicator crossed into heredity. Genomes are historical documents, not sealed manuscripts. The usual story says parasites invade and hosts defend, and that story is true as far as it goes. The deeper story is negotiation over time, because not every insertion remains an active enemy. Many viral insertions are broken, silenced, or mutated beyond function, their genes fragmented into harmless relics. Some persist as regulatory elements, promoters, enhancers, and switches, because viral sequences often carry strong transcriptional signals. Over evolutionary time, those signals can be repurposed into host control circuits. Some are co-opted through exaptation, where selection finds a use for what arrived by accident. A promoter that once drove viral replication can become a tissue-specific enhancer. A viral long-terminal repeat can become a binding site for transcription factors that shape development. The same sequence can be poison in one context and useful infrastructure in another. This matters because it blurs the boundary between attacker and component. A sequence can arrive as an infection and later become infrastructure, and that transformation is not philosophical. It is measurable in gene regulation, development, and immune responses. What begins as foreign can become necessary, and necessity changes the meaning of origin. You becomes less like a singular author, and more like a settlement among competing replicators, stabilized by mutual constraints. The genome becomes a treaty written in mutations. The cell still needs coherence, so it builds defenses that operate at multiple layers. It methylates DNA, compacts chromatin, and uses small RNAs to silence transposons and viral remnants. It recognizes patterns of foreign nucleic acids and triggers immune responses that can shut down translation or destroy infected cells. In the nucleus, chromatin can tighten like a drawn curtain, hiding dangerous sequences from transcriptional machinery. In the cytoplasm, RNA interference complexes can slice transcripts before ribosomes ever see them. Defense is layered because failure at one layer is inevitable. Yet the defenses are never absolute, because absolute defense would also block flexibility. The platform remains porous because it must remain functional, and function requires openness to transcripts, to recombination, and to regulated expression. A cell that sealed itself completely would also freeze its own adaptability. Porosity is not only about viruses, it is also about the idea that heredity must be nucleic acid based, because biology has at least one other way to propagate a message. In the late 20th century, Stanley Prusiner argued that proteins can be infectious, and the claim initially sounded like heresy. The prevailing assumption was that infection required nucleic acids, because replication seemed to require a template of letters. Prusiner's work forced a new category into the conversation. The resistance was not merely cultural, it was scientific caution, because extraordinary claims demand extraordinary evidence, and contamination is always a possibility. Over time, the prion concept survived challenge because it explained patterns that genes alone could not. Prions are misfolded proteins that induce other proteins to adopt the same conformation. The message is not a sequence of bases. The message is a shape that propagates by templating, like a fold that teaches other molecules how to fold. In visual terms, a normal protein is a carefully packed structure, stabilized by many weak interactions. A prion form is an alternative packing, often richer in beta sheets, that can aggregate into fibers. When a normal molecule touches that fiber, it can be coerced into the same misfolded state. Those fibers can grow, fragment, and seed new growth, which is how a shape becomes a replicator. The replication is not of letters, but of architecture. An architecture can be just as persistent. It is a different kind of memory, written into molecular geometry. Prions demonstrate a second channel of biological information, one that travels through conformational state rather than nucleotide sequence. A conformational state can replicate, spread, and persist across individuals, and it can do so without any DNA or RNA inside the infectious particle. It can produce stable phenotypes and catastrophic disease without carrying genes in the usual sense. The result is heredity-like propagation without nucleic acids, and that fractures a common intuition about what information must look like. If replication and evolution can occur via protein conformation, then life is not synonymous with genetics as commonly taught. DNA is a dominant medium, not the only possible one, and dominance is not identity. The category information expands beyond letters into structures and dynamics. The ribosome still matters here, because it shows how blind execution can amplify any replicator that interfaces with the system. A prion does not need translation to copy itself, but it thrives in a world saturated with proteins. A virus depends on translation, but it can rewrite the genome that feeds translation. The cell is a machine that can be used by whatever can interface with it, and that is both powerful and dangerous. The same openness that enables evolution also enables exploitation. The same generality that makes life versatile also makes it vulnerable. Now the tension rises. If the organism is not fully in control of what it calls self, what keeps it from dissolving into noise? The answer is fidelity balanced against change, and the balance is narrower than we like to admit. Replication must be accurate enough to preserve complex organization, yet flexible enough to adapt to shifting environments and new threats. There is an error threshold, a limit beyond which information cannot be maintained. Too many mutations and the genome loses coherence, collapsing into randomness because beneficial structure is diluted faster than selection can preserve it. Too few mutations and adaptation slows, leaving the lineage brittle when conditions change. Life persists in a corridor between stagnation and meltdown. Viruses illustrate this boundary in real time, because their generation times are short and their populations are huge. Many RNA viruses replicate with high error rates, generating swarms of variants rather than a single uniform genome. This quasi-species cloud can explore possibilities quickly, but it lives close to the edge. The swarm can adapt fast, but it risks error catastrophe if fidelity drops too far. Antiviral drugs sometimes exploit this by pushing mutation rates upward, forcing the viral population past its threshold. The strategy is not to block replication outright, but to make replication too noisy to sustain information. Cells face the same logic, stretched across longer timescales and buffered by more repair. DNA repair reduces mutation, but it costs energy and time, and it can slow replication when speed matters. Proofreading is a trade between speed and accuracy, and evolution tunes these systems towards survivable rates under typical conditions. That tuning is not a moral choice. It is a statistical outcome shaped by environments, population sizes, and the costs of maintenance. Even in stable conditions, the cell must budget resources between growth, repair, and defense. This makes life feel less like a protected essence and more like a narrow operating regime. The organism persists because its control loops hold, and because its information stays above the threshold. The unsettling part is how contingent that is, because a slight shift in error rates can turn heredity into static. We can visualize this as a dial on a control panel. Turn the mutation rates slightly upward, and the system becomes inventive but unstable, like a radio drifting into noise. Turn it slightly downward and the signal becomes clean but inflexible, like a locked channel that cannot respond to new interference. Edge cases sharpen the point, because they expose the seams in our definitions. Viruses are inert outside hosts, yet inside they behave like relentless replicators, converting a cell into a factory. Prions are infectious without being alive in the usual sense, yet they spread information through populations with grim efficiency. Sterile organisms are alive without reproducing, yet they are built by a reproductive lineage, and their cells still run the same translation machinery. Each case undermines any single criterion definition, because each isolates one property and shows it can be absent while others remain. So the documentary's central question returns with new weight. What is life physically if its core machinery does not verify meaning? Why are you alive at this moment if your cells run code that can be foreign? The best we can say is operational, and operational does not mean shallow. Life is a process that maintains organized information against thermodynamic drift, in a world that constantly tries to erase structure. It does so by coupling energy gradients to control, copying, and repair. It persists by filtering outcomes through selection, not by guaranteeing truth at the point of execution. The ribosome will translate what it is given. The polymerase will copy with an error rate that is low but non-zero. The organism survives because the whole system over time keeps the errors from winning. This has cosmological consequences, because it ties life to the availability of usable energy and stable chemistry. A universe that can host life must provide persistent gradients, whether from starlight, geothermal heat, or chemical disequilibria. It must provide chemistry rich enough for complex polymers and time for error correction and selection to sculpt robustness. It must allow compartments, catalysts, and cycles to persist long enough to accumulate information, because information is not free. The same physics that drives entropy must also allow local pockets to resist it, at least temporarily, by exporting disorder to their surroundings. Yet even in such a universe, the self remains porous, and porosity is not a metaphor. The boundary of an organism is not only a membrane, it is a negotiated interface between internal control and external code, between what is regulated and what can slip through. Viruses, transposons, and protein states can cross that interface and become part of the story, sometimes as threats and sometimes as tools. The genome is not merely inherited, it is also edited by history, and history includes invasions. So we end this chapter with a more disturbing clarity. Biology can map mechanisms of translation, infection, and propagation with increasing precision, and those maps are becoming almost cinematic in their detail. it still cannot give a complete, testable explanation for why this information-maintaining process becomes you, here, now. We can describe the loops, the enzymes, and the thresholds, and we can even manipulate them. We can watch ribosomes in action, track viral insertions across lineages, and measure mutation rates as numbers on a graph. Yet the lived fact of being a coherent self remains an emergent outcome, not a single mechanism, and that leaves a final observation hanging in the air. If meaning is imposed only after the fact, then self is always provisional. It is a pattern that holds today because the machinery holds today. The question is not whether foreign code exists in you, but how often it has already shaped you. We search for life with instruments trained on a single example, and that example is us. One lineage, one chemistry, one history. Our detectors assume carbon frameworks, water activity, familiar amino acids, familiar nucleotides, and familiar metabolic gases. When a rover sniffs a rock, or a lab tech loads a gel, the same expectation quietly sets the baseline. That tuning is practical, because you must choose what to measure and what to ignore. But it creates a trap. The more specific the assay, the more it can miss biology that violates our expectations. A narrow key opens one lock very well, while leaving every other door untested. Even the word biosignature carries a hidden clause, spoken in the background of every mission briefing. It often means a signature produced by our lineage, using our chemistry, in our environmental range. The signature is not only a molecule, it is an assumption about what counts as a living process. On Earth, that bias hides in routine lab work, where the tools feel neutral because they are familiar. Stains, primers, culture, media, and enzyme assays assume our building blocks and our reaction pathways. A test tube can be an ecosystem with rules already written into the regents. In space, the same bias becomes mission design, budgets, and payload mass. We look for oxygen, methane, complex organics, and isotopic ratios that match terrestrial metabolism. We also look for patterns that resemble our disequilibria, gases that should not coexist unless something keeps pushing them. That logic is sound for Earth-like life, because life is a chemical engine that maintains imbalance. Yet the engine can be built from different parts, and the imbalance could take unfamiliar forms. If life elsewhere uses different solvents, different backbones, or different catalytic motifs, our best detectors might report sterile silence. The disturbing possibility is closer than Mars or Europa, and it does not require exotic planets. A truly alien biology could be here already, coexisting, unrecognized, and effectively invisible. It could be in the same soil, the same seawater, even the same air, present but unread. This is the shadow biosphere hypothesis, stated carefully because it is easy to sensationalize. It proposes microbial life on Earth with alternative biochemistry, sharing environments, but not ancestry. The point is not that we have proven it. The point is that we have not ruled it out with fair tests. The claim is not that evidence is strong, because extraordinary claims need extraordinary constraints. The claim is that our methods can systematically filter out what we do not know to ask. When your sieve is built for 1 grain size, anything larger or smaller can fall through. Most microbiology begins by feeding organisms what we think they eat, because growth is the easiest signal to measure. If an organism cannot use our nutrients, it never grows in our cultures. For more than a century, this produced what microbiologists call the Great Plate Count anomaly. Most microbes seen in nature refuse to grow on standard plates. That anomaly is not proof of shadow life, but it is a warning about method. A microbe can be alive and metabolizing, yet still fail to divide under our conditions. It may need a partner species, a trace mineral, a pressure range, or a slow rhythm that our incubators do not allow. Most molecular surveys begin by amplifying genes we already recognize, because amplification turns faint signals into readable ones. If the genetic polymers differ, standard primers bind nothing and report absence. Even within known life, primer bias can erase whole clades, so a truly different system could vanish completely. Even microscopy can mislead, despite its authority as seeing is believing. A cell-like boundary might not take our stains, and a metabolism might not change the dyes we rely on. Under the lens, a living boundary can look like a mineral vesicle, while a mineral vesicle can look like a cell. A shadow organism could be rare, slow, or metabolically quiet, especially if it occupies a narrow ecological niche. It could persist as a thin film of chemistry that never triggers our alarms. In a world dominated by fast, loud biology, a quiet biology could be overlooked for generations. Some proposals focus on alternative amino acids, because proteins are where chemistry becomes function. Earth life uses 20 standard amino acids in its genetic code, with a few rare additions like selenocysteine. But chemistry allows many more stable variants, and meteorites already deliver amino acids our ribosomes never learned to use. Non-standard amino acids can form peptides and fold into functional shapes, because folding depends on physics, not tradition. Side chains can be longer, more hydrophobic, more acidic, or more reactive. They can also evade enzymes evolved to recognize the standard set, which matters because enzymes are gatekeepers. If a shadow biosphere used a different palette, our proteases might fail to cut its proteins into familiar fragments. Our mass spectrometers might still see peaks, yet our databases would not know what they mean. The data would look like noise, when it is actually a language we cannot parse. Other proposals focus on alternative nucleic acids, because heredity is where life becomes evolution. DNA and RNA are not the only possible information polymers, even if they dominate on Earth. In synthetic biology, researchers have built XNAs, xenonucleic acids, with altered backbones that can still store sequences. Backbones can change while bases still pair, because pairing is about geometry and hydrogen bonding. Sugars can be swapped, linkages altered, and the polymer can remain capable of templating. The result can be an information system that is chemically close, yet enzymatically inaccessible. These ideas are plausible in chemistry, but elusive in practice, and that elusiveness shapes the mystery. A different polymer might degrade under our extraction methods, or never bind our dyes. It might also be insoluble under our buffers, so the very act of preparation erases the evidence. Alternative solvents widen the thought experiment, and they sharpen our awareness of habit. Water is extraordinary because it dissolves ions, buffers temperature swings, and supports a rich acid-base chemistry. But it is not uniquely capable of supporting complex chemistry under all conditions, at least in principle. Ammonia, hydrocarbons, or supercritical fluids can be imagined as media, each with its own constraints. Each changes reaction rates, stability, and what counts as a membrane. A membrane in methane is not a lipid bilayer the way we know it. It is a different compromise between order and flow. We do not need to believe in methane cells on Earth, and the chapter does not ask you to. The point is that life detection often assumes water because we are water. When we say habitable, we often mean habitable for us, and the difference matters. So we change the angle. Instead of asking what life looks like, we ask what our tools are blind to. We stop treating our methods as windows, and we start treating them as filters. The deepest blind spot may be chirality, a property so basic it hides in plain sight. Many biological molecules come in left-handed and right-handed forms, like mirror images that cannot align. They have the same atoms and bonds, yet they do not superimpose, like gloves made for different hands. Earth life is strongly asymmetric, and that asymmetry is one of its most universal fingerprints. Amino acids are mostly left-handed, while sugars in nucleic acids are mostly right-handed. This is called homochirality, and it is not required by physics in any simple way. That choice is not cosmetic, because enzymes are chiral machines built from chiral parts. They bind one hand of a molecule while rejecting the other, not by opinion but by geometry. A binding pocket is a three-dimensional lock, and the wrong key simply will not seat. A protein built from the opposite-handed amino acids would not fit our enzymes, even if it performed similar functions in its own world. Our proteases might not digest it, and our receptors might not recognize it. The molecule would be present, yet it would behave like a ghost in our biochemistry. A nucleic acid built from opposite-handed sugars would not be copied by our polymerases, because the active site expects a specific twist. Our ribosomes would not translate messages written in its mirror code. Even if the bases were familiar, the helix would spiral the wrong way. This is mirror life in its strictest sense, and it is a clean conceptual inversion. It is life built from the same kinds of atoms, but assembled with opposite-handedness. It is not weird chemistry, but a flipped version of chemistry we already understand. Mirror life is not fantasy chemistry, and it is not a metaphor. Mirror amino acids and mirror sugars can be synthesized, and mirror polymers can be built in laboratories. Researchers have even engineered mirror image enzymes, which act on mirror substrates with the same elegance. The question is whether an entire organism could run on that reflection, maintaining metabolism, replication, and repair. If it could, it would be biologically alien while chemically familiar. It would share the periodic table with us while refusing our molecular handshakes. Detection becomes hard for a simple reason that feels almost ironic. Many assays rely on enzymes, and enzymes are the very things mirror life would not interact with. The more biological the test, the more it assumes the same-handedness. A mirror microbe could be present in a sample yet leave no PCR signal, because PCR is an enzymatic ritual. It could be metabolizing, yet fail tests that assume our cofactors. Even a basic assay like does it consume glucose assumes a pathway that recognizes our stereochemistry. It could also fail to grow in our media, not because it is dead, but because our nutrients are the wrong hand. A mirror organism might need mirror sugars, mirror amino acids, and mirror vitamins. In that case, we would starve it in plain sight, then declare the sample sterile. This is where opportunity and threat intersect, and the intersection is uncomfortable. Mirror life could be metabolically isolated, unable to eat our food and unable to infect our cells. That isolation sounds like safety, because it suggests a biological firewall. A firewall exists because our enzymes cannot process its polymers, and it cannot process ours. Infections rely on intimate molecular compatibility, from receptor binding to replication machinery. If chirality blocks those steps, then many familiar risks collapse. But isolation also means our immune systems might not see it, and invisibility is not the same as harmlessness. Immune recognition depends on molecular fits, and mirror proteins can evade those fits. Antibodies are chiral too, and a mirror antigen can be a shape they cannot grasp. Isolation also means our biodegradation pathways might not break it down, which changes the ecology of waste. If our microbes cannot digest it, its biomass could persist. Persistence is not automatically catastrophic, but it changes the accounting of carbon and nutrients. The risk is uncertain, because ecology is not only digestion and infection. A mirror organism could still compete for light, minerals, and physical space. It could shade other microbes, coat surfaces, or clog pores in sediments, altering habitats without ever sharing food. It could still alter environments by producing mirror metabolites, which might be inert to us yet active in its own cycles. It could still change pH, redox states, and nutrient availability. A chemical gradient does not care which hand built it, and ecosystems run on gradients. It could also evolve in its own domain, under its own selection pressures, and that is the deeper unease. Evolution does not require compatibility with us, only persistence and variation. If replication occurs, then adaptation follows, even if the adaptations are unreadable to our assays. The uncertainty is the point, and it is why the idea remains both fascinating and unsettling. Mirror life could be harmless, or it could be disruptive in ways our models cannot easily predict. Risk analysis fails when the object refuses the categories used to measure risk. Now the narrative tightens around a modern warning, because speculation becomes sharper when it meets policy. In 2024, a group of scientists argued that some mirror organisms could be uncontainable. The warning was not about what exists in nature, but about what we might choose to build. The warning, published in Science and signed by 38 researchers, targeted a specific category of engineered organism. It focused on synthetic mirror bacteria built to replicate. Replication is the dividing line because a non-replicating molecule is a hazard you can count, while a replicator is a hazard that counts itself. Their argument was not about malice, and it was not a thriller plot about villains. It was about biosafety assumptions that quietly depend on shared biochemistry, shared degradability, and shared detection. When those assumptions fail together, the safety case can collapse as a system. Containment is a chain of expectations, and chains fail at their weakest link. We expect sterilization to denature proteins, disinfectants to break membranes, and monitoring to detect residues. We also expect that if something escapes, the environment will help us by degrading it. We expect biological controls to work because engineered organisms are often designed with dependencies. We expect engineered auxotrophies, kill switches, and phage susceptibility to limit survival outside the lab. These are clever strategies, but they are clever within a shared biochemical language. But those controls are built from our enzymes and our pathways, and they often use chiral recognition at the deepest level. A mirror organism would not necessarily respond to the same triggers. A kill switch that depends on a protein. DNA interaction may fail if both are mirrored. Standard antibiotics target ribosomes, cell walls, and enzymes that bind our chiral substrates. A mirror ribosome and mirror enzymes could be unaffected, not by resistance mutations, but by geometric mismatch. Even if a drug reaches the cell, it may not dock to its target. Standard disinfectants can still oxidize and damage, because oxidation is not picky about handedness. But effectiveness is not guaranteed. And dose. Response curves matter in real systems. The kinetics and targets can change when the chemistry flips, especially for agents designed for specific biomolecules. Environmental monitoring often uses PCR, immunoassays, and enzyme-based reporters because they are sensitive and scalable. Mirror DNA and mirror proteins might register as nothing at all. In a monitoring dashboard, nothing detected can look identical to nothing present. Even waste treatment is an assumption embedded in infrastructure, not just in laboratories. Sewage microbes break down organic matter using our enzymes, and mirror biomass could pass through intact. The result would not be a dramatic outbreak, but a quiet persistence through systems built to digest one kind of life. The scientist's core claim was structural, and that is why it landed with force. If detectability and degradability fail, then containment becomes guesswork, and guesswork is not biosafety. A system that cannot be measured cannot be confidently controlled. They argued that a replicating mirror microbe could spread before anyone noticed, because early spread can be sparse and silent. By the time you detect it indirectly, it may be established. Indirect detection might come as ecological shifts, unexplained films, or chemical anomalies that appear too late. They also argued that remediation could be uniquely hard, even if the organism is not aggressive. If no natural predators can metabolize it, ecological checks might be weak or absent. A new lineage could arrive without the parasites, grazers, and competitors that normally keep populations in balance. This is the climax of the chapter's first arc, where the mystery turns into a mirror held up to our confidence. The more alien the biology, the more our safety logic becomes a mirror of itself. We realize that many safeguards are not universal, but local customs of our chemistry. So we change the angle again. We move from alternative life forms to the deeper question of how any life begins. Because even if shadow life exists, and even if mirror life can be built, the origin problem remains. How does chemistry cross into evolution? And what is the moment when matter becomes a system that persists? The question is older than modern biology, and it has haunted thinkers since the first microscopes revealed hidden worlds. In the 19th century, debates over spontaneous generation were not only about microbes, but about the boundary between the living and the non-living. Modern abiogenesis research replaced superstition with pathways, but it did not remove the philosophical edge. We can synthesize nucleotides, peptides, and vesicles, yet we still struggle to assemble the whole. The boundary is not a single step. It is a coupling of steps that reinforce each other. This is where Stuart Kaufman enters the story, not as a discoverer of ancient fossils, but as a theorist of possibility. He proposed that life can emerge as a phase transition in networks of reactions. The claim is not that life is simple, but that complexity can appear abruptly once connectivity passes a threshold. The key concept is the autocatalytic set, and it is easier to feel than to define. It is a collection of molecules where reactions produce catalysts that, in turn, produce more of the set. Each part helps make other parts, until the whole begins to sustain itself. In such a network, no single molecule is the whole system, and no single reaction is the essence. The system is the closed loop of mutual catalysis that sustains production. If you remove one component, the loop weakens, but the identity is in the pattern of support. Kaufman's claim is mathematical rather than historical, and that distinction matters for how we judge it. As diversity and connectivity increase, the probability of a self-sustaining network rises sharply. The increase is not linear, because networks have tipping points. At first, chemistry is sparse, like a few scattered roads between isolated towns. Reactions happen, but products dissipate, and nothing reliably rebuilds what was lost. The system forgets itself as quickly as it forms because decay and diffusion erase structure. Then connectivity crosses a threshold, and the map changes character. enough catalytic links exist that the network begins to regenerate itself faster than it decays. A set of molecules becomes a collective, and the collective begins to persist. This is the phase transition framing, and it borrows its power from physics. Like water freezing, behavior changes abruptly when a control parameter passes a critical point. Below the point, the system behaves one way. Above it, it behaves as something new. In water, the parameter is temperature and molecular motion, and the transition is visible in crystals. In autocatalysis, the parameter is catalytic connectivity across chemical space. The crystal is not a shape you can hold, but a regime of self-regeneration you can model. Below the threshold, you have a soup with occasional sparks, and the sparks do not catch. Above it, you have a regime of persistence that can accumulate complexity. Persistence becomes a platform, because what persists can be modified, and what can be modified can be selected. Persistence is not yet life, and the chapter does not pretend it is a solved definition. But it is a new kind of stability, and stability is a prerequisite for history. It is organization that is maintained by internal production, not external assembly. Once such a network exists, selection can begin to act, even before genes look like genes. Variants that regenerate faster or resist breakdown become more common. In plain language, the network that keeps itself going tends to keep itself going. This offers a route from chemistry to evolution-like dynamics without a full genetic system at the start. It suggests that order can bootstrap, building scaffolds that later support templated heredity. It also hints that life might be a continuum of increasing constraint, rather than a binary switch. But it does not solve abiogenesis, and the limits are as important as the promise. It does not tell us which molecules mattered on early Earth, or which pathways actually occurred. A model can show that a kind of transition is likely, while remaining silent about the actual cast of characters. It does not uniquely specify a historical sequence, and history is what the origin question ultimately demands. Many networks could cross thresholds in theory, and only one became our lineage. The others, if they formed at all, may have vanished without leaving a trace we know how to read. It also does not define life in a way that ends the debate, because definitions are tests. It defines conditions for self-maintenance and growth of organization. Yet a self-maintaining chemical machine can still feel different from a living self, especially when we ask about agency and heredity. The central question remains, and it is the question that keeps returning like a tide. Why does a self-sustaining network become a living self, rather than a transient chemical machine? At what point does persistence become lineage, and when does lineage become a world of experience? The open problems cluster around information, because information is the bridge between momentary chemistry and long-term evolution. How does a system store heritable patterns, not just regenerate chemistry in the moment? Without heredity, selection has nothing stable to sculpt. Template-based replication is one answer, because templates can copy patterns across time. But templates need monomers, catalysts, and error control all at once. Without those, heredity dissolves into noise and the system cannot hold complexity long enough to matter. Error catastrophe is a real constraint, and it is not just a metaphor. If copying is too inaccurate, complexity cannot be maintained, no matter how rich the chemistry is. There is a threshold fidelity below which information leaks away faster than it can accumulate. Compartments are another problem, because selection needs units, and units need boundaries. A network needs boundaries to keep products together, but boundaries must also allow nutrients and waste flow. Too tight, and the system starves. Too loose, and the system disperses. Membranes can form spontaneously from amphiphiles, and that spontaneity is one of the hopeful facts. Fatty acids can assemble into vesicles in water, and vesicles can grow and divide under simple conditions. But coupling membranes to replication is non-trivial, because growth must track information. A compartment without inheritance is a bubble, and bubbles are everywhere. A bubble can trap chemistry, but it does not remember what worked. The hard step is turning a bubble into a lineage, where successful internal patterns are passed on. Energy coupling is another problem, because chemistry does not run on desire, it runs on gradients. Reactions need driving forces, and early systems must harness gradients without modern enzymes. They might use pH differences, redox couples, mineral surfaces, or light-driven reactions, but the coupling must be reliable. Metabolism first and genetics first scenarios each face gaps, and the chapter holds those gaps open rather than closing them. Metabolism without heredity cannot evolve efficiently, and heredity without metabolism cannot persist. Each story solves one side of the triangle while leaving the others suspended. There is also the problem of integration, which is where many plausible parts fail to become a whole. How do replication, metabolism, and compartmentalization lock together into a stable triad? The triad is stable only when each part supports the others, and that mutual support is hard to engineer even in modern labs. Kaufman's framing makes a cosmological claim feel plausible, and that is why it matters beyond Earth. If life is a phase transition, it might occur under broad conditions. Broad conditions mean higher frequency, because the universe offers many laboratories. Broad conditions also mean that rare Earth becomes less about chemistry and more about contingency. If many worlds can cross the threshold, then the universe may produce life more readily than we expect. The night sky becomes less empty in principle, even if it remains silent in practice. That sounds comforting until we connect it to the great filter, the idea that something prevents most life from becoming long-lived, spacefaring civilization. If life is common, then the filter likely sits later in the chain. The silence then becomes a warning, not a coincidence. Later filters are darker because they involve complex organization and fragile stability. They include the rise of complex cells, the stability of ecosystems, the emergence of intelligence, and self-destruction. They also include long stretches of time where a single shock can reset progress. They also include technological steps that can outrun control, because technology amplifies power faster than it amplifies wisdom. Uncontrollable bioengineering becomes a candidate because it scales faster than governance. A tool that can rewrite life can also rewrite the assumptions that keep life contained. Mirror life warnings sharpen that tension, because they show how a technical success can undermine a safety framework. If we can build biologies that evade detection and containment, then the filter may be in our hands. The danger is not only what we create, but what we cannot easily measure. The more we raise the probability of life's origin, the more we shift risk toward persistence. Survival becomes the rare achievement, not emergence. The universe may be good at making beginnings, while being indifferent to whether those beginnings last. This deepens the documentary's disturbance, because it turns a comforting story inside out. If life is easy, then your existence is not explained by rarity, but by a narrow corridor of stability. The corridor is narrow because it must satisfy many constraints at once, for long periods. You are alive now because countless constraints are currently satisfied, and because they have been satisfied long enough to produce you. Those constraints include chemistry, energy flow, error control, and ecological balance. They also include the accidents of history, from asteroid impacts to oxygenation events, that could have gone differently. And if other biospheres can exist beside ours, then our story is not the story, but one story among many. It is one lineage among possible lineages. The earth beneath our feet could be more like a crowded library than a single book, with shelves we have not yet learned to read. The chapter's tension peaks here, because the uncertainty is not a small technical gap. Our definitions fail because our sample is small, and our safety fails because our assumptions are parochial. We can map mechanisms with precision, yet still lack a universal account of why this process becomes you at this moment. Transition. We return to the central claim because none of these possibilities yields a satisfying explanation of why you are alive. A structural explanation would have to bridge levels, and it would have to do so without collapsing the mystery into slogans. It would connect thermodynamics, information, and selection to the lived fact of persistence. It would explain how energy flow becomes structure, how structure becomes memory, and how memory becomes a self that continues. It would also have to survive contact with the unknown, because the unknown is not an edge case anymore. It would need to recognize life we have never seen, including life that mirrors us. It would need criteria that do not depend on our amino acids, our sugars, or our enzymes. And it would need to explain why this island of order holds together now in a universe that prefers disorder. The question lingers as the visuals fade. Is life a rare miracle, a common phase change, or a precarious balance that only looks stable from inside? We have toured the edge cases, and the edges refuse to stay still. Viruses blur life and chemistry, inert outside cells yet explosive inside them, where a hijacked ribosome can turn a few strands of RNA into an epidemic of copies. In the microscope, they can look like tidy geometry, icosahedral shells, crystalline arrays, Yet their metabolism is borrowed, rented for minutes from a host. Prions blur genetics, spreading heritable harm without nucleic acids, because a protein's shape can be a template. A misfolded conformation recruits normal proteins into the same twisted fold, an avalanche of structure, not sequence. It is inheritance without genes, and it forces a question. If information can ride on shape alone, what else can? Endogenous retroviruses blur self and other, writing foreign history into your genome, then letting it fade into silence. Over deep time, viral insertions become fossils in DNA, some still capable of awakening, others repurposed into biology's own tools. In placental mammals, fragments of viral envelope genes were domesticated to help fuse cells in the placenta, turning an ancient infection into a developmental feature. Mirror life blurs containment because our enzymes might not see it, and our immune receptors might not recognize its signatures. Swap left-handed amino acids for right-handed ones, or invert sugars in nucleic acids, and the familiar lock and key chemistry fails. In a lab, that sounds like safety. In an ecosystem, it could mean invisibility, an organism that neither feeds nor is fed upon. Phase transition models blur gradualism, because chemistry can tip into persistence abruptly. Networks of reactions can cross a threshold where autocatalysis becomes likely, and once that happens, feedback loops amplify small advantages. The scene shifts from slow accumulation to sudden coherence, like a fog condensing into droplets, or scattered sparks becoming a sustained flame. Together, these fractures point to one conclusion. The category life is unstable at its borders, and our definitions fail where reality is most instructive. The closer we look, the more the boundary behaves like a coastline, detailed, irregular, and dependent on the scale of observation. The ribosome becomes the emblem of this problem. It executes mappings from codons to amino acids with no access to meaning, reading triplets like a punch card machine built from RNA and protein. Transfer RNAs arrive like adapters, amino acids attached, and the ribosome ratchets forward. 3 bases, 1 residue, again and again. It does not verify origin, intention, or future consequence of the protein, and it cannot tell toxin from enzyme, or signal from sabotage. Life persists not because it knows, but because selection stabilizes what works. Over billions of years, blind iteration-tuned error rates, proofreading and redundancy, until good enough became durable. Even the genetic code itself carries scars of history, frozen accidents and constraints, rather than any guarantee of optimal design. So we change the angle. The failure is not one missing fact, but a structural gap between description and explanation. We can catalogue parts and still miss the principle that makes the parts cohere into something we call alive. Single criterion definitions collapse under edge cases because one property can vanish while the process continues. Metabolism can pause in spores, reproduction can be outsourced in viruses, autonomy can be partial in symbiosis. Multi-criterion definitions become circular because the criteria are chosen from known life, then used to certify that same life as the standard. Operational definitions become tool-dependent because instruments encode assumptions about chemistry and history, and what we measure is shaped by what we expect to find. We can list components, map pathways, and simulate fragments, yet still lack a universal boundary. A cell is not a checklist. It is a dynamical regime, a pattern that persists through flux. What looks like a stable object is really a maintained process. Molecules replaced, membranes renewed, signals corrected before they cascade into failure. What we call an organism is porous code running on a thermodynamic gradient. Identity persists through turnover, repair, and error control, while intrusion remains constant. Viruses entering, plasmids swapping, microbes negotiating at every surface. The system is historically contingent, patched by evolution, and never fully sealed. Even your mitochondria carry the memory of an ancient merger, a bacterial lineage folded into the architecture of your cells. Now we widen the frame to cosmology. Life exists because the universe supplies long-lived gradients, from starlight to geothermal disequilibria, and because planets can keep those gradients running for eons. Sunlight drives photosynthesis, chemistry drives hydrothermal vents, and both create pockets where order can be paid for with exported waste heat. The cosmos also supplies chemistry that can store information in polymers and compartments, because carbon bonds flexibly in water allows molecules to meet, separate, and meet again. Those conditions can host life, but they do not uniquely entail you. A habitable zone is not a destiny, and a rich periodic table does not promise a mind. Even if the steps are mechanistic, the path is contingent, one branching history among countless unrealized alternatives. Even perfect mechanism does not answer why you are alive right now. It explains how your loops run, not why this particular loop became a self. At some point, description becomes a mirror that reflects processes, while the question keeps asking for a reason the mirror cannot provide. The great filter returns with sharper teeth. If origins are common, then the dangerous steps may come later, in complexity and technology, where feedback becomes planetary. Synthetic biology can create new replicators, including mirror lifestyle systems that evade detection and degradation. And the risk is not malice alone. It is mismatch, an entity optimized for persistence in a world whose defenses were never built to see it. Uncontainability becomes a cosmological risk, not just a lab risk. A single runaway replicator could rewrite ecosystems, and in the long view it becomes one more candidate for why the sky might be quiet. The same creativity that lets intelligence understand life also lets it manufacture new edge cases. So what is the honest answer? You're alive because an evolution-stabilized information system is currently maintaining itself against entropy, and it is doing so in a narrow corridor of energy and error. It exports disorder as heat and waste, repairs damage before it compounds, and copies patterns within an error budget that is neither perfect nor random. The miracle is not that it never fails, but that it fails slowly enough to persist. Yet we still lack a complete predictive theory for when chemistry must become life. We can propose pathways, estimate probabilities, and build partial models, but we cannot yet point to a planet's chemistry and say with confidence, here, life is inevitable. The boundary is not a line we have found, it is a line we keep drawing. We also lack a definition that survives all possible biologies. A full account would unify information, thermodynamics, evolution, and emergence into one operational test, and it would remain valid when the substrate changes. It would tell us what to look for when life is not made of our familiar parts. Final beat. Our best science can describe the machinery that keeps you alive with breathtaking precision, from ribosomal kinetics to membrane potentials to the statistics of mutation. The disturbing part is structural, because alive still has no final universal boundary. The universe may not respect the boundaries we wish it had, and it may be teaching us that the deepest categories are not nouns but behaviors. Across this journey, every clean definition of life cracked, because the thing we call alive keeps slipping between categories and edge cases. What held steady was a deeper pattern, matter organized into loops that harvest gradients, export entropy, and spend energy to preserve information. In cells, the ribosome reads any compatible message without meaning, while repair, proofreading, and selection, plus viral intrusions and prion shapes, negotiate what counts as self. Our instruments inherit that bias, so a shadow biosphere or mirror life could sit beside us, chemically real yet methodologically invisible, and the great filter may hinge on whether replication can be contained at all. So why are you alive right now, in this exact moment, if physics can describe every part without naming a reason? The unsettling answer is that science can map the mechanisms of this thermodynamic rebellion, yet it still lacks a substrate-independent test for why one precarious process becomes you. Maybe life is not a substance you can bottle, but a temporary victory condition, an information pattern that keeps paying the universe's entropy bill. And if the cosmos offers only gradients and time, then your existence is not guaranteed by law, it is negotiated, second by second, against collapse. If we want to recognize life elsewhere and survive our own creations, we'll need better questions, better detectors, and a humbler theory of what alive really means.